What's that?
We're starting on, but we're finishing Leishmania, right?
Yep. Yeah, we've got one more species to talk about. All right. Fire weather out. That's what my desktop says. There's a don't start fires. All right. Had a question about exams. Our exam is scheduled for the last week of April. And then that first Monday of May, that's our lab practical. So that's the last week of lab, we'll have our practical. Next week's Good Friday. It's not a holiday for us, but it possibly could be. So on Thursday, it's happened a couple years. I can't remember if they did it last year or not, but email went out like Thursday afternoon, and the president announced that Friday a holiday, campus holiday, so no classes. So just be flexible. If not, we'll still have class on that Friday. The following Friday, I am out of town. It's a lecture day, so I think what I might do, we'll see how far along we progress and whatever. It may be another presentation that you'll watch and then take a short quiz on it to learn another group. But we'll see. We'll see what we get through this week and then next week. Or we'll see what we get through next week and then like the Monday, that following week. Don't forget, some of you have presentations on Monday. Make sure you get those to me before lab. All right, we left off here, Leishmania. We had 3 species that we're talking about. Leishmania tropica gives the cutaneous form of leishmaniasis. Leishmania donovani, which is more of a visceral form, goes into organs and then can come back as this post-colozoidal leishmanoid. And our last one is here in South and Central America, Leishmania brasiliensis. This one is a bit interesting because it usually starts off as cutaneous leishmaniasis. So you have your lesion that forms someplace on the body, and it's going to occur typically where you had the bite. If left untreated, that scar that lesion will heal. That will heal. Same thing, if you get treated, it'll heal. However, if it resolves on its own, or if it is inadequately treated, meaning you didn't get the full run of the treatment, then this parasite will come back as mucocutaneous leishmaniasis. Mucocutaneous. So this is the mucous membranes is where it'll come back. And you're not going to get lesions on those mucous membranes because you don't have that skin. It's mucous membranes. And what you have is just significant damage and destruction of those parts. This has been called espundia or uta. There are several other names depending on which country in Central and South America we look at. But these are probably the 2 most common names that you might encounter that describes this form. So this mucocutaneous form will cause degeneration of cartilage and soft tissue, usually of the nasal and buccal mucosa. So normally it comes back on the face, someplace on the face. And this is the result. You have complete destruction, degeneration. This is the nasal septa that gets destroyed. You have parts of the nares destroyed. You can have the hard or the soft palate, the hard and soft palate, mainly start to get destroyed. Destruction of the lips and mouth. It's all degeneration. Complete destruction. Now it can develop quick, so you can get the lesion and then it can start developing almost immediately. Or it may take, as I said, up to 30 years later after that initial lesion heals. But you have this transition basically from the cutaneous lesion form to this mucocutaneous form that leads to a lot of disfigurement. Relapse is common. if it's not treated properly. So I knew a graduate student when I was in grad school. They did work down in the Amazon. He came back with a classic lesion on his forearm. This is the first time I've learned how our country handles some of these diseases. The doctor could not prescribe the meds. It had to be first diagnosed by the CDC, and then the CDC then allows them to prescribe their meds. They prescribed it for like 7 days, but when you look in the textbooks, they all say, so you've got to prescribe it for at least 10 days, 10 to 14 days. So we asked about it, said you should contact the doctor. This isn't the right prescription, the right length of time. At least not in the literature. It's not the right length of time. Don't know if he did or not. I think they may have said, don't worry about it. They cleared it up, and literally it was a month or two. It came back mucocutaneous form, came back in his nose. His nose swelled up. I mean, he looked very swollen. And that's when you had to go through that whole process again. The CDC had to diagnose it in order to administer the drugs. And at that point, he had to have a drip. So they inserted a drip on his arm so it could be continuous treatment. And then, they resolved, but he's still missing part of his nostrils. Don't know if he's probably missing parts of his septum too. But mucocutaneous form, not something you want. Difficulty with this parasite is that it does utilize many different reservoir hosts. For example, sloths and anteaters, we know, serve as a reservoir host. You don't really see these types of destructive infections in these animals, but they're there. It's keeping it out in the environment, so you're spreading via the sandflies that are down there.
Can you go back one slide?
Donavani? We did. Yeah, and then the other one was Tropica. So location, and it is. The location kind of determines what types you might encounter. There are other Leishmania species here in South and Central America, but Braziliensis is usually the one that you'll hear about. Any questions? All right. That's bad for the disfigurement. So next up, so I do have a quiz that's posted. Apparently I don't have a life cycle quiz, so that's what I got sidetracked today. I was going to work on it. I got a different idea on how to do the life cycle quizzes, so I think you're going to be the guinea pig on them. But we'll get those posted probably by Monday, and then again you'll have, I think, 3 attempts to kind of practice on those. But we're going to move on because we are not done with the flagellates yet. We're not done with the flagellates. So there's 2 more groups that we're going to talk about, the Retortamonada and the Parabasalia. All right, and we have specimens of these. So we're going to start with the phylum Retortamonada. This group has several features. So number 1, they lack mitochondria, and they lack Golgi. Now in this group, the Golgi are called dictyosomes. So they don't have them. What does that mean for our organism? What does that mean? Okay, so they'll have to, if they're going to do oxidative phosphorylation or something like that, they're going to have some other way. All right, what else? What are Golgi? Dig deep. I think some of you are seniors, right? Have you taken that field assessment test yet? It's a good review. Yeah, so like processing and then secretion of different proteins and molecules and so forth. Right, so yeah, it's a feature of Retortamonada. This group also possesses recurrent flagellum that are in a cytostomal groove. So cytostome, it's like a cell mouth. You've encountered it. Where would you have encountered it? Maybe in paramecium. I'm trying to think when I first encountered it. But it's a groove is what it is. It's not a mouth, it's a groove, and that's where our flagella, sometimes of our flagella can rise. So the recurrent flagella means that you have it originating maybe near the anterior end, usually they are anterior end, and they should progress forward, but they instead loop back and start pointing posteriorly. That's what our recurrent flagellum is, and you can see several of them. For these, you have your cytostolon groove. So instead of coming out of the body and then being moved back, you have your basal body extending the flagella, and then the flagella runs through a groove or through a channel in the body going out to the exterior. We're only going to look at this guy, Giardia. So they have this recurrent flagellum that points to the back. It rises anteriorly and then curves back to the back. And then these guys occupy anoxic environments. If you don't have oxygen, then there's really no point to have your mitochondria, right? So that's why they lack it. All right, so in this phylum, we have a specific class that we're going to look at, class Diplomonadia, and the order Diplomonadida. All right. In this order, members of this order, they have two identical nuclei lying side by side, eyes, and you're going to have 1 to 4 flagella per nucleus. All right, 1 to 4 flagella per nucleus. Ready? All right. So our species is Giardia duodenalis or Giardia lamblia. So these are same names; they're synonymous. This was first observed by Antony van Leeuwenhoek in 1681. Where did he find it? In his own feces. That is dedication, right? What's looking for it? Or? Well, I was just gonna ask, where have you've probably encountered Leeuwenhoek before? Yep. Yeah, it was entirely microscopes. Yeah, he kind of put together his microscopes and started looking at cells. You're right, started with the environment and then went to bodily fluids. You're starting to look at things that are microscopic, and of course he decided to look at his own feces. Dedication. I bet his family was very proud of him. All right, especially if it was in like the dining room with this. Thank God. Anyways, yeah, he described it. Described this parasite, and he saw this, and I believe he also saw the cysts that were there. Now, you had several species, so people were giving this Giardia different names. So we had duodenalis, we had lamblia, we have Giardia intestinalis, but sequencing has really kind of shown that no, these are all just the same species. So they synonymized them. I would do duodenalis or lamblia. Either of them are going to work. I think now they're leaning towards just duodenalis. And it's a cosmopolitan parasite of humans. It is more common in warmer environments because it's more likely that you're going to be able to shed these things out into a aquatic environment and then have people accidentally ingest the cysts. All right, ready? There it is. It is the most common flagellate of the human intestinal tract. We can detect it in about 80% of raw surface water. So just go out to a lake, stream, probably have a high chance of detecting it. Prevalence of infection, though, varies by region. Some regions pretty high, other regions not so much. You can still detect it, but not so much. And again, a big part of getting this parasite is you have to, if you get one, even just one cyst, possibly you'll get it, but more than likely you have to ingest several of them. So like you'll get it, but you're not going to really show any sort of pathology. In order to get that pathology, you're going to have to consume several of these different cysts or ingest several of multiple cysts, say 100, 200 cysts, and then you'll start getting pathology. We'll talk about that. Wild animals may serve as reservoir hosts, and I mentioned including beavers, because the pathology of Giardia, so we call it giardiasis, or this is also the causative agent of beaver fever. So you'd have individuals going out and hiking or camping, and they get to a beaver dam and they drink the water and they get sick. Ready? All right, life cycle. Look at this. I got more chalk. Not only that, I also picked up some colored chalk that I could add to our diagrams if they work. Oh no. We'll see. I got rid of all that other crap. Should check this. Guess we'll have red out here. All right, let's do a life cycle. And I did leave spots for it. Life cycle isn't terribly complex. There are some key points that we'll talk about. So, where are we going to start? Giardia duodenalis. Yes, we will start here. We will have a mature cyst, and we'll add description to it. But our mature cyst is going to be consumed by our host. Right. Our host is many different types of hosts. Both humans and other mammals. All right, so we consume this mature cyst, and then you will have what is technically an excysted stage. All right, so with this excysted stage, we know it's different because it has 4 nuclei. Two sucking discs. Adhesive discs, probably. 2 sucking discs. 2 locomotor apparati. Which are basically this region where all of our flagella originate. There's two of them instead of one. So it's basically double. And then what's going to happen, once you get excysted, we will complete division to produce our trophozoite stage. All right? This is going to occur, or trophozoite is going to be in the small intestine. So what is a trophozoite? What does that name mean? based on its, break the word down, troph. What do you think that means? Trope. How do you call it? What's that? Almost. Energy is a good guess. Where do we get our energy? Food. Some of you have heard trophic levels. All right, that's our food levels. Trophozoite is basically our feeding stage. And I'll put in quotation marks. We're going to encounter this in these life cycles. So the trophozoite is basically the standard stage. That's what Giardia is. That's what we're going to see. We are in the small intestine. That trophozoite is going to replicate by binary fission. And it's this stage that's going to be responsible for all of our pathology. We'll talk about that. Now, they are in the small intestine. But as they replicate, population size is getting larger, and then they will slowly move down into the large intestine and the colon. All right? When they transition here, now we will start to have early encystment stage of our trophozoite. Early encystment. So at this stage, our cyst wall is being secreted. So we're going to start changing our shape to be more oblong shaped. And it starts secreting the cyst, and then it will be, the cyst wall will be finished being secreted, and we will now have a young cyst. And this young cyst will have two nuclei associated with it. Two nuclei, your one, you can't see your adhesive disc, you won't see it. You'll only have one locomotor apparatus, and so forth. All right, this cyst will then continue maturing to get to this mature cyst. This has our 4 nuclei. Two sucking discs, 2 locomotor apparati. All right, and then this cyst gets shed out into the environment. I'm gonna write that. That gets shed up into our environment, and then you get consumed. But that's not all. That's not all. Because it doesn't just have to be the mature cyst. It doesn't have to be the mature cyst. All that we need is that cyst wall starting to get secreted for our stages to start getting secreted. We're passing with the feces. That's all we need. So we can have young cysts come out of the host. We can have any stage in between that young and the mature cyst leaving the host, or we can wait until the mature cysts come out. You are going to see a range of ages of these cysts that will come out of the host and be able to infect our individual. But we have to get to the mature stage in order for it to be infectious. So this is our infectious stage. That's what's going to get consumed by our next host. Trophozoites are going to be coming out too. They're going to, when you see our pathology, you're going to have a lot of trophozoites coming out into those species. That's how Lowenhoek was able to identify it. But those won't ever develop into a cyst. They'll just die. So what are some of the triggers here that we see? Well, what is this first trigger? We're moving into the colon. Obviously, something's happening here in the colon that's triggering this encystment, because you don't see it in the small intestine. we saw it in the small intestine, we'd say, yeah, we have this encystment starting. So what do you think the trigger is? What's that? Good guess. It's not pH. pH is part of this life cycle. Where can we see a change in pH? This cyst knows it needs to start existing when it goes through the stomach, and it's the pH that triggers it. Since people have collected these cysts, isolated them, exposed them to bile salts, that's not it. They expose them to maybe some movement, that's not it. It has to be the low pH. Has to be the low pH of the stomach. So that's where pH comes into play. Good guess. What else? What happens in the colon? What's that? Okay, liquid gets absorbed. That's what happens there. Is that the trigger? No. We have that. So if you go back to some of our life cycles, we've talked about that seems to be a trigger. Once you start getting into the colon, the drawing out stimulates formation of the transformation of some of our life cycle stages. That would be the logical thing, right? That's not what's actually triggering it. What triggers it is cholesterol deprivation. So our body is going to start taking up some of these fats and lipids. And as we progress, it'll have fewer and fewer cholesterols. Trophozoites, they sense that and say, oops, I must be moving into a lower place. Let me start encysting so I can prepare for life outside. So in our trophozoite has two nuclei. They have two nuclei. That is our character. Those are the eyeballs of our Giardia. And then you have binary fission. That's how you replicate. And then our mature cyst has 4 nuclei. And we'll see some. You can see some in lab. You'll have to be up at oil immersion in order to see some of the nuclei, and you'll have to fine focus. There's a lot of them. You'll have 2 of them up near the top. You'll have those in focus, and then you have to fine focus and move down. So those will be out of focus, and then you'll see 2 more cysts or 2 more nuclei behind them. All right. That is our life cycle. Not terribly difficult, right? All right. Some key points for the life cycle. This is a monocytous life cycle. So single host, that's all it takes. Humans, other animals, again, a bunch of other animals can serve as a host. If you just kept with humans, that works. We'll go for it. Excysment, triggered by low pH of the stomach. So uptake of water in the colon, and then your cholesterol deprivation. Cholesterol deprivation really tends to be that key. Because again, you can kind of get these things raised up, and once you start losing that cholesterol, then they start going for encystment. Our young cysts have 2 nuclei. The mature ones have 4. That's what you need to get. And then a single stool could have up to 300 million cysts in them. It's a lot of cysts entering the environment. And you can have up to 14 billion parasites in a diuretic stool. Diuretic. Yeah, diuretic. I was going to say diuretic. It's like diuretics. It's like makes you pee. I think that's what a diuretic is. Diarrheic. Yes, this will cause diarrhea. It's a real good watery stool caused by trophozoites. 14 billion, massive. So cysts are immediately infectious once they come out, assuming that they are your mature stages, right? If they are your mature cysts, they're going to be immediately infectious. You can start getting infected, but we may need some cell division to form trophozoite. So that last step, once you come out, finish division to get our trophozoite. And we do have some SEM images just so you can kind of show your recurrent flagellum. Definitely, it's in a cytostomal groove, right? You don't see the flagella out. You do have some that run on the sides. So this is the bottom. That's our adhesive disc or sucking disc. And then you have your dorsal side of the organism. Ready? Let's talk about this. I think I have immune evasion next. Yep, immune evasion. Very fast replication of trophozoites happen in the host. You get there, it'll be, this isn't waiting a week. This is, they start replicating once they exist. And that means then that the cysts are going to start to be shed before the host even mounts an effective immune response to it. So I'm going to say this is an immune evasion response mechanism where you basically get in and get out before the host responds. Right? You do things super fast to avoid that immune response that wipes you out. If left untreated, it'll take about 2 weeks for you to clear it. And this is, your body will produce antibodies and will produce a defense and will help to take out this parasite. But in some cases, if you have immunocompromised individual, those are pretty susceptible. If you have some other factor that hinders your immune response, that infection could become chronic. And this is strain specific. So some strains, I'll use myself. Maybe I don't know if I've ever had it. Who knows? But if I had it, perhaps, today I get it and I clear it out in 2 weeks and we're good. But then a couple weeks later, I go out and I get it again, different strain. Maybe it becomes chronic where it's not this massive type of diarrhea, but it's regular bouts every week or every 2 weeks or so, where it lasts for a day or two and then it kind of clears up. That's where it says it can last for months with frequent relapses, and usually these chronic forms aren't as bad as like the one-time deal that we typically get. Immunocompromised and children are at high risk of this getting chronic form. They're also at high risk of the parasites itself. You'd be surprised. Really, in children, it's about dehydration. That's the issue that we often see with the children. And immunocompromised. Ready for the pathology? No gross images. All right? Oh, hold on, before we do that. The other thing, I forgot about this. Actually, I didn't. I thought about it just as I started talking. But we do have some antigenic variation that gets exhibited. CF compared to the trypanosomes. So we talked about the VATs, VSGs. Kind of a similar idea. This Giardia can express up to 180 different antigens over the course of 6 to 12 generations. And these different antigen types vary in their infectivity and the variance, like how much pathology they actually cause. And this is kind of a great example to demonstrate this. So they did immunofluorescence. They're chasing it with monoclonal antibody against various antigen types. And at the first point, you're basically only getting one antigen type here. But after several generations, so this is all green, and sorry, it's red and green. That's not my choice. But after a couple generations, now you start seeing, you still have some of the green, but now you start adding some of the red fluorescence showing up, indicating we have the second antigen type that has appeared later on. This could be a way in which we could bypass some of the antibody response and cause the infection to persist a little bit longer. Because as we know about the immune system and the immune response, you're going to develop an pretty strong antibody response in a couple days. All right, you're going to be there, and the infection should probably clear out in about 5 to 7 days, but it lasts 2 weeks, and perhaps this is why it's just some of this shifting of the antigens. So you kind of have a couple different pathways. Now, we're ready for the pathology? Yes, now. Numerous names, all refer to Giardia. It's all related to consumption of water. No big surprise. The cysts need water. They'll dry out. So, beaver fever, that's one. The wilderness trots, that's number 2. Hiker's disease, number 3. Kind of get to this idea that it's hikers. I don't want to say survivalists, but individuals that go out. Why would that be the case? They're drinking. Why would they drink water that they find on the ground? They rough it. Has anyone been out and gone on a hike in mountains or something like that? You've seen movies like that. If you haven't, just imagine yourself. I've hiked in North Carolina in the Appalachian Mountains. You get up to it and you see this mountain stream, man. Crystal clear water running down, moss-covered rocks. I sat on a massive boulder sitting on the moss as cushioning. The water just looks so great, and it's like nothing's contaminating it. Wrong. Giardia. Yeah, I drank water out of a Canadian lake. Avoided it. Kind of went out deeper water to get it. But yeah, that's more than likely what happens. Our systems, our sanitation system is going to clear it out. Even if you filter it, have a water filter. Water filters are going to remove these cysts. Not all of them. I don't think. I'd have to double check those Brita filters, but tap water, yeah, it's clear. So what causes it? What causes the pathology? It is the attachment of trophozoites to the intestine. Those trophozoites have a sucking disc, an adhesive disc. They're going to get in there. They're going to get onto the intestine. And what's going to happen is they're going to cause inflammation and then ultimately villus atrophy. So death of the microvilli. It's this irritation, inflammation that starts to then increase mucus production that leads to a decrease in absorption by the small intestine. So the small intestine should be absorbing stuff, should be absorbing things from the food. But as soon as you start stopping that absorption, it stays in the intestine. So it's going to be passed down. So you can start to see we've got mucus, increased mucus production. You're going to start having epithelial tissue slough off of the intestine, all this stuff. You've got still the contents of the intestine that isn't going anywhere. It's still getting moved down, ultimately producing this part down here. So your explosive and watery diarrhea. You're going to have the foul-smelling stools called steatorrhea because of the fat absorption. You're not uptaking the fat, so you'd have even more fat in the stool than what's typical. And associated then with all of this, you're going to have just a general malaise, your general not feeling good, abdominal cramps. Again, it's irritating your intestine. Weight loss, because you're not really going to be absorbing nutrients. There's inhibition that's happening. And then, of course, flatulence, excess gas, because bacteria are just going to be like, oh, plenty of food, plenty of gas, and so forth. It's not anything close to Leishmania. You can get through it, but it could be just have some major discomfort. Can we treat it? Yeah. Yeah, you can treat it. Dogs can get it. I think that's where, has anyone's dog had it? That's people that I knew in Memphis, that's where it was their dogs. How do their dogs get it? How do we get it?
Is it the same way? They just drink water?
Exact same way. Where do you think they're drinking their water? Yeah, maybe a little puddle, but who has a dog? Have you guys been to the dog park? Oh, dog parks. All it takes is one dog out there to contaminate the entire pond. That's all it takes. So, yeah, that's usually when people are like, yeah, I've heard of Giardia. Oh, you did? Did you have it? No, my dog had it. Same thing. Yeah, explosive watery diarrhea. And we now have a dog park. Maybe a second one. Any questions? All right. Next phylum, Parabasalia. So I don't know if you've tracked how many phyla. We're now on our third phyla for this section. This is Parabasalia. Members of this phylum possess parabasal fibers that originate at the kinetosome. These parabasal fibers form what's called the parabasal body, the fibers are associated with this body called the parabasal body. That parabasal body is the Golgi complex. But again, when descriptions were being made, I mean, think about Van Leeuwenhoek. He had no idea. We didn't know mitochondria. We didn't know what Golgi were. So people started giving names to different things. So yeah, in this group, the parabasal body is the Golgi complex. And it's associated with the fibers. We'll see it. Or we can see it depending on our specimen. This group also possesses an axostyle. Some would call it an axostele. The axostyle is non-modal, so it's not going to move. It's just a stout median rod. Its function is to serve as support for the individual, for that cell. Instead of mitochondria, they possess hydrogenosomes. Right, so hydrogenosomes are double membrane-bound organelles that produce energy via substrate-level phosphorylation. Right, so mitochondria, we're not going to have the electron transport chain, but we can still get some of the benefits of the electron transport by using substrate-level phosphorylation. Not as good, but it's possible to use it. Now, they got their name hydrogenosome because they utilize protons for basically the proton to hydrogen to do the redox balance, the reduction oxidation balance. And they use hydrogenase to do so. So these hydrogenosomes, again, special. Can we see them in our specimens? I'm going to say no, we can't really see them in any of our slides, but we can identify them on diagrams because the location of these things could be diagnostic. They could be diagnostic, and you'll see some of the diagrams. The Parabasalia could have thousands of flagella on them, all right, and they are all flagella. And most of our members are symbionts of animals. So you could have possibly seen a slide called termite symbionts. I think this would have been in zoology. You would have seen a bunch of these flagellates, and a lot of them are in the phylum Parabasalia. All right, so we've got the parabasal body, hydrogenosomes, axostyles. Ready? The group of parasites that we're going to talk about are the Trichomonads. This is a class Trichomonada and order Trichomonadida. The morphology is general. So we have an anterior tuft of flagella, we have an axostyle, and we have an undulating membrane. But there's a kind of a lot of variation on what we see and how we see it. So just within a species, there's a lot of individual variation. So in our organism, this is it. Our anterior ends up here. You've got your tuft of flagellas. You usually have an Ungular membrane, that again, a flagella runs down that length. You have your axostele, that's our stout median rod. We can see this, it stains darkly. You've got your nucleus that's there. The parabasal body is associated with the parabasal fibers, and your kinetosome complex. So this, I believe, came from Wikipedia, possibly. But there are a lot of things that we can kind of see. So the parabasal fibers are these guys that are in the interior of our organism. We do have one other thing. That is the costa, which is this dark structure here. It seems to be associated with the ungular membrane, or forms the base of the ungular membrane. And the possibility of this thing is that maybe it provides some sort of resistance so that the undulating membrane, if this flagella whips, you can get some undulation that allows movement of these guys. So members of this group are found in the intestinal tract. Some members are found in the reproductive tract. And this is in both vertebrates and invertebrates. So there's 3 species of Trichomonas that are kind of important for us. So Trichomonas vaginalis, this is in the reproductive tract of humans. Trichomonas gallinae, this is in respiratory tract of birds. And Trichomonas tenax is in the mouth. This is in the mouth of humans. So gum lines. We don't have any specimens of tenax. We do have specimens of vaginalis, and we have specimens of gallinae because we had one of Dr. Skipper's graduate students did a survey of this in the pigeons, and she found quite a few. So we're going to talk about these 3. These 3, yeah, they're going to differ. We do have, I think, these notates of the hydrogenosomes, like where they are. Sometimes some of them run just the length of the axostyle. Some of them run where the costa is. Some of them are just scattered all throughout. So probably not going to see them. But we're going to stop here. We'll pick this up on Monday. We'll probably finish this, and then we'll move to the amoeba, I think. Yeah, we'll talk about the amoeba before we hit our apicomplexans. All right? All right. Y'all have the quiz. I made it until Monday evening. So, oh, man. This is a pain. I should have checked these. All right. Y'all have a good weekend.